with introducing our speakers. So our first speaker this morning, Dr. Volker Gerdts. He is the director and CEO of VIDO, which is the Vaccine and Infectious Disease Organization at the University of Saskatchewan in Canada. Volker is from Germany, and he trained at the Hanover Veterinary School and also Federal Institute for Animal Health in the island of Reims in Germany. And his research interests are in the area of vaccine development for humans and for animals, with a special interest in swine and working with PRRS, PEDV, and pseudorabies. Volker was unable to join us in person, but we welcome him warmly from Canada with a new post-election result, and we look forward to learning more about vaccines. So I'll transfer to you, Volker. Oh, sorry. We should have one more slide here. Yes, Monty always keeps me on task. We welcome your questions, both through the chat and through text, and the number for texting the messages, the questions is up on the slide, so thank you.
Assuming you can the audio guys just confirm that this works right now? All right, well, good morning, everyone. It's great to be here, although virtually. I'm sorry about that. It's very complicated right now with travel going back and forth, even despite the fact that we're so close, really, in between Minneapolis and Saskatchewan. Congratulations to Egan and congratulations to Casey. That's both well-deserved. So what I wanted to do this morning is talk to you about vaccines. And so what I'm gonna try is to combine a little bit the basics of vaccines with maybe more newer concepts like the RNA vaccines, for example, and use both some of our important pig diseases as examples, so African swine fever and PRRS and so on, but also then to make it a bit easier for everyone to understand, come back to COVID-19 a few times so that now that everyone is expert on vaccines, make it maybe hopefully easier to understand for everyone. So just a little background here. As you heard in the introduction, I'm the director of VIDO. VIDO stands for the Vaccine and Infectious Disease Organization. We're located in Saskatchewan, as you can see here, so just north of you, across the border. We're part of the University of Saskatchewan. We have scientists from more than 25 different countries represented. You can see there 45 plus PhD scientists. Overall, we have more than 160 staff members. And over the years, commercialized a number of vaccines, all of them animal vaccines. But now with this current pandemic going on, we actually have a COVID-19 vaccine and clinical trials in humans. And we're also working on a number of other human vaccines that are going forward. So VIDO's niche is really in this interface between human and animal health. And that's, as you know, we often find emerging diseases originate from more than 75% of all pathogens or new emerging diseases that have originated over the last few decades, all have come from animals and jumped from there into humans or vice versa. And so this is where we do this important work. This is Canada's largest high containment lab. And this is what is called a BSL-3 egg facility. So you can see here on the right side, that's where the labs are. And then on the left side is our animal wing. And in this, we can house over 1,000 pigs under BSL-3 egg conditions, which makes this facility one of the largest in the world, in fact. But you can see we're not only working with pigs in there, we're working from very small animals like bats, as you can see over here, to very large animals. We just completed a study in bison, looking at tuberculosis in bison. And so it's a very versatile facility, specifically designed or mainly designed for the use of large animals. We do have an international board of directors, and I just wanted to point out two individuals that you all may recognize. So Amy Cronin, our vice chair, from Cronin Farms, operating in Ontario, Iowa, and Missouri. and then Rory McAlpine, former senior vice president with Maple Leaf Foods. Both are very active board members and representing the pork industry on our board. Now, I don't want to go through this, but I just wanted to highlight what I just said. Vito is working both in the human and the animal health area, but we also work on interesting pathogens like these protein misfolding diseases, chronic wasting disease, which seems to be in the news again. Vito had a vaccine for chronic wasting disease, which unfortunately didn't give us the level of protection that we were hoping for. And so right now we in fact working on a second generation vaccine for it. But you can see for pig diseases, there's a number of them that we're focusing on here. And of note, we're one of the few labs in the world that are able to work on African swine fever. And so we're using adenoviruses as a viral vector for this disease and working on a vaccine right now, but also on other approaches that are not vaccine-based. But I was asked today to talk to you about vaccines. And so I'm gonna try to do a little bit of both. Tell you in principle how vaccines work and what in principle the technologies are, and then bring here or there a little example in trying to make a point, and then at the end come up with a little summary. So starting out, in my mind, this is what the ideal vaccine looks like from a researcher perspective. And of course, that would hopefully then also apply to you and how you use them. We want these ideal vaccines to have a fast onset, meaning ideally after immunization, days after, and we all witnessed this with the pandemic. You don't want to wait for months and months for the immune response to kick in. Ideally, you want this to happen within 1 or 2 weeks, or if you can push it even faster than that. And at the same time, we want to have a long duration of the immune response. Again, COVID is a good example. We don't want our immune responses to drop after 2 or 3 months. We want them to ideally last, a year, 2 years, 5 years, 10 years, or, as we see for some of our vaccine, even for lifelong immunity. Ideally, vaccines work after a single immunization. There's a few on the market, as you know, in pigs that are working after a single immunization. That's a big challenge, though, and I think we will in the future still have to work on that to make that possible. We want them to be highly effective. We all are now math experts when it comes to percentage. We know that our COVID vaccines are all 93.6 or 94.1% effective. But really what we want these vaccines, we want them to be overall highly effective so that when we use them, we get good protection, not only against clinical disease and clinical symptoms, but ideally also against transmission. And that is something that is very difficult to accomplish. And of course, very much depends on the pathogen that we're trying to protect from. We want these vaccines to be prophylactic and therapeutic. What do I mean by that? Well, prophylactic is the traditional use of vaccines. We use them in healthy animals before the disease is there. And then when the disease arrives, these animals are protected. But we also would like our vaccines to work in the face of a disease. So being therapeutic, meaning you have already your barn broke from a disease and you go in and you vaccinate. And so all the animals that haven't been infected yet are responding and are protected, but also the ones that have been infected are dealing with the disease right now. The vaccine would support what their immune system does and protect them or help them to protect even better. We also want our vaccines to work in all age groups. So that could be from very young to very old. And in particular in humans, in niche populations, but also, in animals in niche population, those specific groups or breeds that immunologically are slightly different, and we want them to work in there. Of course, we want them to be safe, no side effects, and we want them to be easy to store, transport, and administer, and also easy to manufacture, which reduces the cost, which for you is probably the most important item on the list here. We want these vaccines to be cost effective. Now I'm putting on another thing down there. We want them to be a DIVA vaccine. And so that's a concept that is coming up now, of course, but with African swine fever, everybody is aware of the trade implications of these diseases. And when you have them, how you lose markets and so on. So the future is really for vaccines to be DIVA compatible. And what that means is simply shown on this very simple graph here. There's a lot of text here, but essentially what these vaccines have is a marker in them that allows you to, through a companion diagnostic assay, to determine if the animal has been infected or vaccinated. And so the very basic concept you can see here, so we have a structure, this could be a pathogen, a virus in this case. These viruses have lots of proteins on their surface that are being recognized by our immune system, and the pig is making antibodies to them. As we know, the spike protein for COVID, that would be one of those examples. So if we can create a vaccine that lacks one of these structures, as you can see, so the red one, then the pig wouldn't be making a red antibody because there is no such structure. It doesn't recognize them. And if there is no red antibody, then you have a diagnostic test that specifically measures whether the red antibody is there or not. If it's there, you know the animal has been infected with the wild type. If it's not there, you don't know if the animal was vaccinated or not, but for sure it hasn't been come in contact with the infected or with the wild type virus, and which is why these assays then have two proteins. So one that allows you to say it has been vaccinated, so black in this case, and red is missing. So the combination of black positive and red negative would tell you that this animal has been vaccinated but not infected. So very simple procedure. It's molecular, not very difficult to create vaccines that lack these marker proteins. We have now a number of them coming forward. And in the future, much of our trade will be based on these companion assays that allow us to differentiate vaccinated from infected. Now, when we talk about infectious pathogens and vaccines in general, there is really a number of pathogens that we want to protect against by using vaccines. And so it starts with parasites, which we all know, we can see they're centimeters long. Some of them, the intracellular ones, the amoeba and so on, are much smaller, but many of these parasites are very large. And so the immune system uses very unique mechanisms that allow us to protect us from these extracellular pathogens, whether it's being worms that live on the surface of our intestinal cells, or even arthropods or ticks and insects and so on that live on the surface of our skin. So the immune system has mechanisms in place that specifically allow us to address them. And that's the goal then for our vaccines, to activate those specific mechanisms. Now, when we go into bacteria, we're now in the micron size. So bacteria are typically between 1 and 2 micrometer in size. They come in all different shapes and forms. They live within our cells, or they live on the outside of our cells, so intracellular or extracellular. And again, the immune system uses very different mechanisms to attack them. And then the last one, of course, as we all know very well right now, are these viruses. They are very, very small. So the coronavirus, for example, that causes COVID-19, a good representative of it. And they vary. The smallest ones would be in the 60 to 100 nanometer range, and the largest one would be in the 150 to 250 micrometer size. So if you just take an average virus and put it on the row, on the rim of a quarter as shown here, you can have 5,000 viruses sitting in a row. So it just shows you how small these pathogens are. And it's amazing how much impact they can have, as we all witness as part of the during pandemic. Being small, though, allows them to easily spread. And this is, of course, a big concern to the industry, as we all know. So they can be transmitted via droplets, as you can see. And I tried to find a pig here, but really these studies have been done in humans. And you can see all these individual droplets when we sneeze that are coming out, and each of them containing many of these virus particles. But then, of course, we know via body fluids or on trucks, for example, that we use to transport, those are very common mechanisms by which these viruses spread from one barn to another. And air is often involved in this as well. And that is why when we work with them, we have to protect our staff. And so you can see on the right side how we need to make sure that our staff does not breathe in these pathogens that they can't see, they can't feel. They're essentially invisible to our eyes. And so as you can see here, They are carrying a backpack on a small unit, on a filter unit on the back, which blows HEPA-filtered air away from their face into the suit here. But really, we're working with these viruses in what is called a biosafety cabinet, a piece of equipment that creates an air curtain right here. And so you reach into it, you do the work, but the air curtain prevents the virus from spreading. And so this is important because this is, like, I think the global pandemic right now shows the need for these facilities like ours and many others in the US that are equipped to work with these pathogens. And they're equipped to make sure that nothing will ever leave this facility, nothing will ever get out. But at the same time, our staff is fully protected, especially if these are human viruses. And so what happens then when we breathe this in, or when our pigs are breathing this in? It's the same principle. And I purposely use here both humans and pigs interchangeably because I wanted to show you that much what we hear in the news right now about vaccines and so on is also applicable to pigs. And in fact, veterinary medicine, veterinary vaccines are light years ahead of human vaccines when it comes to innovation. Most vaccines, more newer technologies, most innovative technologies have been used in animals before they actually go into humans. And so these viruses then attach to the cells, as you can see here, they use structures on these cells. So this is now in your nose. This is one layer of epithelial cells, we call them. The viruses come and they make their way into it. And from here, then they use the cell to make lots and lots of copies of it. And from here, they spread to other sites in the organ. And so the pig doesn't look any different, essentially has the same structures in the respiratory tract as the humans have, the same tissues, the same type of epithelial cells. And most of our infections occur either in the respiratory tract or in the enteric tract. More than 99% of all infectious disease and more than 95% of all infectious diseases are actually beginning at the mucosal surfaces. And so that is a reason why in the future we will see many more vaccines being delivered via the mucosal sites, because we want to specifically target the mucosal immune system, what's called the mucosa-associated lymphoid tissues. That's where we want the vaccine to be initially induced, and that's where we then get most of our responses. So once these viruses have made their way in, it's shown here. So you can see this is a cell, what I just showed you there. This would be the cell surface here. And here in green are all these virus particles. They have been stained in this case, so they're easier to see. But you can see these virus particles are now trying to attach to the cell. And once they're attached, they're going to make their way into the cell, into the inside of the cell. And that's now where viruses take over our cells, the cell factory, and they make millions of copies of themselves. And so this is shown on this little graph here on the right side. You can see the virus needs to attach to the cell to then inject its RNA into the cell. The RNA, one of the nucleic acids, so there's RNA and DNA viruses, so they carry the two different types of nucleic acid. They're injecting it, and now they're hijacking the cell machinery, taking it over, and now instructing it to make lots and lots of new copies so that they can spread to other sites within the organism, to other organs, but they can also spread into the environment and infect other animals and so on. And I wanted to point out one thing here that often in the media we hear a lot of misinformation about. You can see this RNA here. So in this case, it would be COVID-19, an RNA virus, and the virus injects its RNA into the cell. And this happens all the time. We come in contact with the virus all the time during a viral infection. And so you can see here that what we are so concerned about RNA vaccines injecting RNA into our bodies, this happens all the time when we have RNA infections or RNA viruses infecting us. So the RNA itself is not a dangerous molecule. It happens all the time. And so these long-term consequences that people are concerned about with these RNA vaccines, you can see here that this is a natural process that all happens all the time. Now, what are the mechanisms of how our immune system can protect us from it? Well, one is to make antibodies that prevent the attachment of the virus to the cell. And so the viruses are using receptor structures on the surface that they hijack and that allows them to bind. If we have an antibody that binds the outside of the virus, as shown here, it can now neutralize the virus, meaning it no longer can attach to the structure on the cell surface. It no longer can go into the cell. And the virus on the outside of the cell, it doesn't matter to us because they're not able to survive there for long. They need to get into the cell to make copies of themselves. And the other mechanism our immune system uses are what's called T cells, specific immune cells that recognize once a cell is infected with the virus and says, in order to deal with this, it's easier if we just kill the whole cell with the virus inside rather than trying to depict it and specifically attack the virus. And so T cells are called cytotoxic T cells coming in and they destroying the whole cell. And with it, they also destroy the virus that's on the inside. So it's easier for the immune system just to get rid of the whole infected cell rather than specifically trying to attack the virus. And so how does the immune system actually see these pathogens? I showed you how big they are or how small, in fact, they are, while the immune system sees only structures, only bits of them. So it's even smaller than that. And so on the surface of these viruses, I'm going to go back here. On the surface of these viruses, you can see the structure here, which is called, in the case of coronaviruses, the spike protein, and which is what most of our vaccines are targeting right now, because we want these antibodies to prevent the attachment. While Jason McLellan identified the structure of the spike protein, and you probably all have seen this in social media and so on, pictures of it. This is what the spike protein looks like. It's a trimer, 3 of them are coming together. And when it attaches to the cell, this part here flips over, it changes slightly this conformation, and then it allows the virus to attach. Now, this is still not how the immune system recognizes it. Our immune system only recognizes certain peptides, certain areas as shown here in red of this whole protein. And that's what we call epitopes, and antibodies are binding to them. So to the immune system, it's an agglomerate of epitopes presented in a certain structure, which now allows us to make these antibodies and to recognize it. And T cells, in fact, don't even use the whole structure. T cells get these small peptides presented to them, and they're only that long, so 10 to 15 amino acids in length. So why am I telling you all of this? Because it informs you how we make vaccines and what we show the immune system, and that certain criteria are important when we want more B cell responses, so more antibody responses, or more T cell responses. Now, all of our vaccines are designed to give us immune responses and last for a long time, as I mentioned. And so I'm just trying to use a very simple graph here. In fact, I use this for my veterinary students all the time, trying to explain it to them. So if on the y-axis we would have our immune responses, or down here we have time, and then I just randomly decided here, this dotted line, that's protection. So if we're above, if our pigs are above, their immune system protects them from the disease. If we go below, they're not protected. So normally what happens is the first exposure takes place, our immune system responds, makes an immune response. You can see for a while we're above the line, our animals are protected. But then when the pathogen is gone, we don't need to carry this on for much longer. So we let it drop again, and we're below the protective line. Now, when we come in contact with this pathogen again, this time our response is much faster, much higher, much longer lasting. And you can see we have a much bigger area above the line, meaning our pigs are protected from this particular disease. And this is what is called immune memory. This is what we call a secondary or an anamnestic response. This is because our immune system has created a memory that allows us, when we come in contact with this pathogen again, to now respond faster and more effectively. And so you can see here an anamnestic response, and that is related to immune memory. Now, this is the principle then for how we use our vaccines. So what we do is we use a vaccine in mimicking a first exposure to this pathogen, as you can see here. And now when our animals come in contact with this disease, it would be really for them the first time. Our immune system thinks it's the second time we come along, And now we're making this very strong immune responses as shown here. That's the principle of vaccination. That's the principle of how we use these vaccines. And you can see we're not using, or most times we're not using the real pathogen anymore. We only use parts and structures of it, and we call them antigen, material of these pathogens that are being used to induce an immune response. Now you can do this multiple times and in fact drive the response higher and higher until you reach a plateau when a boost really doesn't make any difference anymore. Multiple exposures are not increasing the response much more. In fact, you might even run the risk of depleting certain aspects of the response. Now, this is how vaccines are normally being used or were used, but we have evolved from then. And so these days, we're really using vaccines more like this. We don't even want to wait for memory to kick in, although we could, but it would take days for the secondary response to kick in. What we want these days is essentially give us long-lasting protection that hopefully last longer than the life of our pigs. And so they would be completely under this curve, would be completely protected for this time. And so we don't have to worry about the response ever dropping below the line anymore. So the criteria that are important in vaccine development are onset, duration, and then also overall the type of the response. And so when you now boost, you can see you can use the booster even when you are above the line. And so you're essentially just bringing it up again and buying yourself more time, extending the duration of the immune response. But the point is that I'm trying to make on the slide, these days, we usually don't let the response drop below the line of protection, although we could because we have memory. It's just more effective to keep it up there and so that these animals are always protected. Now, there's various types of vaccines on the market for pigs and also for humans. And so this is a little summary of what we have there. And then I want to go with you through each of these technologies and in principle show how they work and use some examples of pig diseases as well as COVID, trying to explain how they work. So the first ones to start are really the live vaccines and the inactivated pathogens. And so here in this case, I specifically picked COVID-19, and so there is very little live attenuated vaccines, but I'll come back to that later on. So for a live vaccine, you would have, you would have the whole virus here, and it's still live, but it's attenuated. It doesn't cause any disease. Now for COVID, we don't have live vaccines, at least not in the Western world, in North America. So what we have and what some companies are working on is an inactivated virus. So in this case, you would take the whole structure, inactivate it with a chemical, or you could also use heat and then use it as a vaccine. And so in this case, the immune system sees multiple structures, as you can see there. So the spike protein, but also all the other structures, and we're making an immune response against all of these other structures. DNA or RNA vaccines, they're now focused only on a gene that is encoding for a specific structure. And so, for our RNA vaccines, it's the ones that are encoding for the spike protein that are currently in development. So the Pfizer and the Moderna vaccine, for example, they have the RNA, the piece of RNA that encodes for this specific structure here. And so in this case, we're injecting the RNA, as I alluded before, and now the cell is making the spike protein for us, and the immune system then sees it and makes an immune response against it. You can also use a vector to deliver this, and there's my Live-attenuated. I'm sorry about this. There is a vectored vaccine here that delivers the same gene of interest, now using a virus as a delivery vehicle to inject the DNA into it. But it's the same principle. We're just injecting either RNA or DNA that encodes for one protein only. So for a subunit in this case, it's the trimer here, and we're using a vector to deliver that nucleic acid. Or in the case of the Moderna vaccine, we're using the RNA directly, which is protected by a lipid vehicle that carries it and allows it to not be destroyed by the body. And lastly, we can make what's called subunit protein vaccines. And those are essentially just making the protein in a manufacturing facility and injecting now the protein here. And then we're getting an immune response to the protein. And that's what, for example, the Novavax vaccine or the Sanofi vaccines are doing that are coming on stream now. So live-attenuated vaccine, as I mentioned, we're injecting a live pathogen in this case. It's grown in cell culture. In most cases, it's attenuated. It's our responsibility in the lab to ensure that it's attenuated and doesn't cause any disease in our healthy animals. It may sometimes cause disease in immune-compromised animals, which is why we don't use live vaccines in pregnant sows, for example, because there is a risk of either causing disease or, for example, getting into the fetuses, into the unborn pigs. Now, the advantage of these live attenuated vaccines is that they very much mimic the natural infection, and therefore they're giving us very good immune responses. So they're very effective, long lasting, give strong antibodies responses, strong T-cell responses. So they are the ideal vaccine. Is that really true? Well, not quite. The disadvantage of them is that they are also shedding. And so we can have the vaccine virus now circulating in our population. And there is always a concern that because of the trade implications, we lose our ability to trade with other countries. So think about ASF, for example, African swine fever. You do not want to have a vaccine virus circulating in the American or Canadian herd. We'll never get that under control again, and it will take away big markets for our industries. So a live attenuated vaccine is good for some diseases, but for others, it's simply not an option. The advantage, though, of these technologies is that they're giving very good immune responses and typically also long-lasting immune responses. And so here's an example of the ASF vaccines that are in development, and you know there's a very promising candidate coming out of Plum Island right now. The issues are still how to grow it, but it seems like we have maybe found a way of how to grow the vaccine virus. And so this is what is called these double deletions or triple deletions, live viruses that we have modified, or these researchers have modified to not be as disease-causing anymore. We call that virulent. So they're attenuated, and they're giving us very good responses, which for ASF, that's a big challenge. And we need to have a very good immune response, both antibodies and T cells, to protect against this disease. Now with ASF, the problem of these live vaccines is that we still seems to be that they're not well adapted at the moment. So if you don't give enough of them, the response is too weak. If you give too much of the vaccine, if the dose is too high, they may actually cause disease. And so the Plum Island one looks very promising in that it seems to be that it is well attenuated. So even at higher doses, it doesn't cause disease in these animals. But of course, shedding of the virus still remains. So from a trade implication, that's a big concern. And then of course, the other question is how stable are these attenuated viruses later on? Can they change? Can they become suddenly a new virus that has kicked out certain elements out of its genome and now behaves differently? And so here is just as a point to highlight this. So you can see the Plum Island vaccine, the I177. You can see it lacks a deletion here. So there is a part that was knocked out of the genome. And because of that, the virus is now much more adapted, doesn't cause disease, and seems like to be an effective vaccine. But this can also happen in nature. And so you probably have heard about this. In Germany, there is a vaccine that is now being under development on reams. And that was a vaccine that was attenuated in wild boar. So in these animals directly, the virus evolved and suddenly came up with an attenuated version that seems to protect as well. And you can use it, as you can see here, for even oral immunization. So my point of this is these live attenuated vaccines are lacking certain parts of the genome. They are attenuated and not causing disease anymore. And if we do, and if we can prove that they're stable, they're very, very good vaccines. And so even in humans, there's many of our vaccines, measles, for example, and so on, that are live vaccines because they're giving us very good responses. Now, if we want to go with a safer option, we use inactivated vaccines. And so inactivated vaccines are grown essentially the same way as a live vaccine. But here now, we're destroying the pathogen at the end of the manufacturing process to ensure that it's not live, that it's dead, and so that it does not cause disease anymore, has no potential to transmit or even get into unborn animals. And so you can see here in this case, we're injecting now dead material into these animals. But because it's dead, they're not very good in inducing immune responses. And so what we want in this case is to add adjuvants or other molecules to them to stimulate their response. Now, the advantage of inactivated vaccines is they're very safe, of course. There's no reversion to a virus that can cause full disease. There's no shedding, and overall, inactivated vaccines cannot cause disease. The immune response is not as good anymore. We get strong antibody responses, but we usually get only weak T cell responses, which makes these vaccines less effective, and which is why we add molecules to the vaccine that we call adjuvants to make our vaccines more potent. And there is a whole area of research in immunology on identifying the best adjuvant and you know, driving the immune response, driving the type of the response, whether this is vaccine for cancer versus a bacterium versus a virus versus, for example, an allergy. So all of this can be modulated and driven by the choice of adjuvant. So that's an inactivated vaccine. The next development then was to now go with what we call subunit vaccines. So rather than using the whole pathogen, in this case, we only now using a certain structure of it. And this is shown here. This is, for example, an influenza virus. So rather than using the whole virus, we're only using now one of the molecules, the hemagglutinin in this case. And as I explained before, either we use it as a protein itself or we deliver it via other methods. And so this is very easy to manufacture now. You can make it in bacteria, you can make it in yeast, you can make it in insect cells. So from a manufacturing point of view, these are easy to make. And so in there, you find now the actual subunit protein, so the antigen, or you can have one or multiple in there. And that's a big advantage of these vaccines. Then you have these adjuvants that are modulating the response, as I just mentioned. And of course, as vaccines include, there's also diluent stabilizers and preservatives. So when we inject our subunit vaccines, now you can see I don't have the whole virus here anymore. It's just a structure now. This is also dead. It doesn't cause disease. So it's very safe. There is no shedding into the environment. It's very cost effective to make. But the immune response, again, is not as strong as a live vaccine. We're getting good antibody responses, but our T cell responses are weaker, which again is why we're using these adjuvants to drive the response and make them more potent. And so, for example, for African swine fever, that's now one way of going down that direction. People have used what we call protein cocktails. So many proteins from the virus mixed together and hoping that we can enhance the immune response that way. It looks like when you inject them just as a protein itself, for ASF, that didn't really work. But what worked is when we now use these protein cocktails and we use vectors, viral vectors like adenoviruses, to deliver them together. And so the latest data from Pirbright actually looks very promising that these protein cocktails may work in this case. And so what they're using are viral vectors that allow us now to specifically deliver these structures, as I explained before. So in this case, we're using the genes that are encoding these structures, and we make a new virus that expresses them. And so I'll explain this here. So we can use a normal virus of our pigs or of humans, adenoviruses that by themselves essentially cause very mild disease, so they're not very harmful to our animals or to humans. And then what we can do through using molecular technologies is put into this adenoviruses now proteins from other viruses. So in this case, these two influenza strep units that I show you here that are encoded, let's say, on the surface right here. So what we do then is we create a virus, an adenovirus, which now carries, in addition to its own protein, also these proteins from the influenza virus. So you have now a recombinant adenovirus that expresses influenza proteins on its surface. And so this is the same as our Johnson & Johnson vaccine or AstraZeneca vaccine. There we're using an adenovirus that is carrying now the spike protein for COVID-19. So we can make these viruses, they're harmless, they don't cause disease, and we use them as a delivery vehicle for these proteins that we're really interested in, the influenza or the COVID proteins in this case. And so the advantage of them is that it's a live pathogen, so it gives us a very good immune response. Itself, the vector doesn't cause any disease. So the responses are very long lasting. They're giving us good antibody responses, good T-cell responses. The advantage is they cannot revert back to virulence, but the disadvantage is that we are also getting an immune response against the vector. And so therefore we can't use this for many immunizations, for many boosters, which is why we sometimes have to switch the vector. So it is possible that you could, for example, use a porcine adenovirus in your first immunization and then use a bovine adenovirus in your second immunization, and you can circumvent this problem. And so this is a technology that we can see now a lot with these viral vectors. And so, you know, essentially what we're delivering there is the gene, which then encodes the protein on the surface of the structure. Now, the last technology is our RNA vaccines. And of course, everybody these days is an RNA vaccine expert. What we now are doing is we're just injecting the gene, encoding the protein on the structure. So this gene here that encodes for these proteins, and that is now being injected. It needs to go into the cell first to make this protein, and then the immune system recognizes it. It's a very effective response. It also gives us long-lasting immune responses, good antibody responses, and good T-cell responses, which is why these COVID vaccines are very effective right now. The advantage of these technologies is that there's no live pathogen involved. It gives you both T-cell and B-cell responses, so antibodies and T-cells. But the disadvantage is that the delivery remains an issue. And that really was the biggest discovery in the last year. The Canadian company who developed a lipid carrier for these RNA vaccines. And now we found a way of how we can deliver at least the COVID vaccines in humans. This is going to be the issue in the future in animals. We need to find ways of making these RNA vaccines more stable and have them easier to use in a barn. We can't have ultra low freezers sitting in every barn for people to use them. And so this is where the future is going to focus on making these RNA vaccines more widely available. So are they the future? Well, they allow for sure rapid development. And so if we need to be able to respond rapidly to a new disease, RNA vaccines offer a lot of advantages, not only development, but also on how they're being manufactured. The other advantage is you can combine many of these RNA molecules. So, you know, not only variants of concerns for COVID, for example, but you can throw multiple pathogens in it, whether it's different strains of PRRS, whether it's a combination of influenza and PRRS, you can combine these in these RNA vaccines. But as I mentioned, at the moment, they're more complicated to handle, transport, and store, which makes them impractical to use in animals. But I would predict that that's just a matter of a few years before we have those problems solved. The RNA itself is very unstable, so you need to protect it. And a better way might be to use DNA vaccines, which is a very similar principle, but offers a few more advantages in terms of stability. Now, other factors that I didn't talk about today is what we are working on is how can we enhance the duration of the immune response so that we essentially have lifelong immunity, or maybe 15 years or 20 years in humans, and pigs for sure lifelong. I mentioned the type of immune response is very difficult or very important to induce. And it varies a lot between bacterial and viral vaccines. I just brushed over all of that, but we are now able to make vaccines for cancers, for example, which require completely different immune mechanisms than making vaccines for E. coli, for example. As I mentioned, mucosal immunization is going to be the future, although very difficult in pigs to do, but everybody's working on it. That needs to be done because the most effective part of our immune system actually sits along the mucosal surfaces, and that's where we want our vaccines to work. Neonatal pigs and so on. Cost, of course, important. And then, as I mentioned in the beginning of the DIVA. Now, one of my last slides here is the mix match of vaccines. And so that is something that is being discussed now publicly a lot around for COVID. And so the quick summary here is, from my perspective, most vaccine types can be combined, and there is a lot of research in the literature that shows that if you prime, so the first shot, if you prime the immune response with a vaccine that gives you a very broad response, or ideally something that induces both T cell and B cell responses, you can almost boost with any other vaccine afterwards and still enhance the response. And so it really seems that the priming, the first shot, is critical for the outcome. So there you want to use something that induces a very broad immune response, like a live vector or an RNA vaccine, for example. And then you can, for example, boost with a protein or a subunit vaccine. So here's my summary slide. Vaccines remain the most effective means of controlling infectious diseases. Of course, there's many alternatives, whether they're drugs like antivirals or small molecules. There's many, many new vaccine technologies in development, and I'm very happy to state this again. It's in the animal vaccine development where we're seeing real innovation happening, and from there it spills over into humans, including the RNA vaccines and DNA vaccines. To just put this in context, I'm now in my 50s. My PhD was on a DNA vaccine, which is essentially the same as the RNA vaccine, and that was done in pigs first. So again, innovation happens in our veterinary medicine and then spills over into humans, and we will see many, many more vaccines coming out in the future. And here's the last little bit. This is where I think the field is taking us. We will no longer wait until a disease breaks out, as you can see here, and then catch up with those diseases and make vaccines, which traditionally for humans takes 10 to 15 years. For animals, takes about 5 to 7 years. Now, if you throw a lot of money at it, we can maybe bring it down to 2 years, as we've seen with COVID and with the RNA vaccines coming up. The real question for us is, why do we always have to wait until a new disease emerges? Why can't we make vaccines in anticipation of the next disease outbreak and have these vaccines made today and tested today so that we're ready for the disease of tomorrow? And I would argue now that the science is there to do this, and this is what many of our researchers at VIDO are focusing on. So with that, I'd like to thank you for your attention, and I'm happy to answer any questions that you might have, either live or on the chat. I don't know how the organizers are doing it, but I'm sure it will work somehow. Thank you so much.
Thanks very much, Volker, and we still encourage people to submit questions if they haven't. We have several here for you, Volker, and again, thanks very much for a very detailed and comprehensive overview. Question here is that you talked about the need to have more mucosal vaccines. Do you foresee the development of a mucosal vaccine for PRRS virus that would be highly effective and broadly effective, I think?
I do. Like, the problem, of course, with PRRS is that we need to know what to put in the vaccine to get protection. And so many of us have worked on the various antigens that PRRS has. So PRRS is a very small virus. It only has a few proteins that you can use there. But in the long term, for PRRS and for many other diseases, we need to get better ways, or find better ways to induce mucosal immune responses. That's where most of these viruses enter our organism. That's where, in fact, the largest immune compartment sits. And so the more or the better we can stimulate the mucosal immune system, the better we protect our pigs from future diseases.
Thank you. Another question that may be vaguely related, but with respect to African swine fever virus, do we have a thorough understanding of the immunogenic epitopes, and why can't we make a safe and highly immunogenic vector for mRNA or vector or mRNA vaccine for African swine fever?
So to put this in context, if you think of the PRRS virus, you might think of a scooter. And if you think of the African swine fever vaccine, you need to think about a Boeing 737 or something like that. African swine fever is such a big virus which carries so many proteins and so many structures. Half of them, we don't even know what they do. And so the challenge for us right now is that the African swine fever brings mechanisms with it that allows it to evade the immune system, to essentially go undetected, to modulate and shift the response, to even turn off specific immune functions. And so with a vaccine, the first thing, you need to know what you're using as a vaccine. You need to know what is the correlate of protection, we call it. You need to know how the immune system controls that particular disease. And for African swine fever, we simply don't have the knowledge at the moment. So what people are using right now is, as I mentioned, a cocktail of different antigens. And now you can use RNA for that. And those experiments are underway and it may work. But at the end of the day, the problem with African swine fever remains that it's a very, very complicated virus. And we need to first understand the virus before we can have effective vaccines. And unfortunately, that knowledge is not there at the moment.
Thank you. Question about whether you have any experience with immune complex vaccines and their use in the swine industry as are apparently used in the poultry industry. And I can't clarify that one much for you.
Did you say complex?
Yeah, immune complex.
Oh, immune complex. So, yeah, so that's another way. That's what's called more passive immunization. So you actually give antibodies rather than giving the actual antigen. That is possible. So that's more a treatment strategy. It's being used in humans and children, for example, that are being infected. And we also, of course, use it during COVID, as you know, the antibodies that are being developed. You can use that in poultry when you essentially use the poultry antibodies. So the antibodies that are in the egg to protect against the disease. That is possible, but we don't really use a lot of it in pigs.
Thank you. One more question. Is there any instance in which live attenuated vaccines being used with adjuvants would be warranted, and could you improve weak T-cell responses of inactivated vaccines using an adjuvants that stimulate the Th1, Th2 immunoresponses?
Yes, to the latter. So inactivated vaccines rely on adjuvants. So all inactivated vaccines that are currently on the market for pigs have adjuvants with them. Now, live vaccines don't really need more adjuvants because they're bringing their own natural adjuvants. So these adjuvants are really what we call danger signals to the immune system. And these live vaccines, these live pathogens carry their own danger signals. So you don't really need to further adjuvant the responses there. So we normally use adjuvants only in inactivated vaccines or in protein subunit vaccines.
Okay, we're tight on time, but I'll ask one more question that's from the list here. Could you comment briefly on what has rendered development of a HIV vaccine near impossible in the human medical area?
So you were breaking up there for a second, but I think you were asking, why is it so difficult to have a vaccine for HIV?
Correct. Yes.
So there, the key is, again, this is a virus for which the mechanism of how it causes disease is the Achilles heel of the immune system. So the virus specifically targets what we call T helper cells, specific immune cells, and by depleting them, we essentially take away the ability of the immune system to respond to it. And so there's a combination of things that we need for HIV, antibodies, and we have identified now the structures for which they encode for these antibodies. So we know that we can protect or we can, like vaccines that induce high levels of antibodies are very effective, but you also need very strong T-cell responses with them. And so that's the challenge there for HIV, to find the right type of vaccine that gives you both at the same time, preventing the virus from invading, and then when the virus gets in, it essentially takes out the main mechanism of the immune system, and so therefore makes your vaccine very, very ineffective.
Thank you very much, Volker. I ask everyone to thank Volker for sharing his vast knowledge of vaccines with us. So again, thanks and farewell to you, Volker. Hope you can stay on.